next speaker. I think many of you already know Lei Zhao, who taught us during our online school in October. And so Lei is a postdoc with Rasmus Nilsson and Torfinn Corneliussен in the University of Copenhagen, and Lei will works with genotype likelihoods and all this interesting representation of uncertainties. And today he will tell us about novel and fast phylogeny inference methods that allow for uncertainties introduced by sequencing and power coverage. So Lei, you're welcome to start.
So can you hear me?
Yes, and we can see your nice slides.
Yes, thanks for inviting me here. And today I want to discuss a little bit on the phylogeny reconstruction method. But as I focus on the ancient DNA stuff, so actually I will not touch the fancy stuff as recombination as Professor Malinsky just said. So I will start my talk. So the phylogeny inference actually now have been widely used across different subjects of biology. It can be applied to learn the evolution history of focal organism. It can also learn the relatedness between different species, and it can also locate it where am I, or where our species are at this moment. But even though it is very popular, the current phylogeny inference method do not keep pace with the state-of-art sequencing technology. So today in the talk, I want to draw your attention to this issue and introduce two new methods. Yes, so let's first, I actually list several of the modern or common ways for people to construct the phylogeny. And all of these methods actually require you input some kind of a very long sequence. We can call it the representative sequence of a sample. And when we put the sample together, we can infer a distance or genetic distance or divergence time between different samples. And we use that distance to infer either a likelihood tree or a distance, pairwise distance tree. But they don't actually tell you how to get this very long sequence. So basically, I listed maybe 4 different strategies to generate this kind of long sequences. Maybe some of them are still used by current biologists. The first one is for each of the sample per site, you just randomly choose one nucleotide rate, and you can reconstruct this kind of a pseudo haplotype genome. The second one actually is per site, you just choose the consensus rate of the genome, and you construct this kind of genome, very long sequence. And the first one and second one actually lack the information of haploidy. So you can imagine that if you get this kind of sequence, actually you don't know whether it is a diploid gene or whether it is a multiploid gene. You just randomly choose or maybe consensus choose a rate per site. And so people try to do, actually they argue the first two are not that perfect. So they try to do something called ambiguity GT. So basically this means per site, you maybe according to the genotype likelihood or other things, you choose the most probable genotype. And if it is a homogeneous site, most probable genotype is a homogeneous site, then actually you can use ACGT to represent it. And suppose it is a heterozygote site, and they will use this UPAC codes and to treat them as either 50% of A, 50% of G, then you just treat them as a R, and you will construct your sequence. And at this specific site, you have a rate called R, and you can construct this kind of sequence. But all these four methods actually just allow you to use one sequence per sample to infer the relationship. If our sample is from the modern like NGS data, actually we will have a lot of short rates, and the short rates will be piled up like along this reference genome. And because of this base calling error, some of the actually rates are not that accurate. So you cannot just randomly choose one, or you consistently choose one to infer to use them to represent the sample. And if you do so, especially when the base calling error level is high, or if the rate depth is high, actually you cannot give a very accurate inference of the divergence time or genetic distance between samples. So people start to thinking about that, and they try to use different kind of methods like measurement of the uncertainty. They incorporate that into this kind of phylogeny inference. And one way, actually, that is the only way we know how people use it, is this NGS-based method. And this method actually is incorporate the genotype likelihood into the phylogeny inference. And they try to say that because they introduce this kind of uncertainty measurement, they can get a more accurate result. But as I will present later, actually, this method also not that good. And that's why I actually want to introduce our own solution to this modern NGS data. So actually, we have two solutions. The first one is called descends the genome. So this one is assumed that actually we have already know the algorithm we try to infer is a diploid algorithm. And this is easy to extend to like maybe multiploid organism, but we just stick to a diploid one. And because we have like per site, we actually have a lot of observations. Like maybe suppose you have rate depth 5, and for this site, actually you will have maybe 5 rates. And the summary is we still use genotype likelihood, and we use the genotype likelihood and observation to infer posterior genotype distribution. Actually, it is a joint genotype distribution. That means it is a genotype distribution of both samples. So the first element of this matrix actually is the chance the first sample at some site is AA, and at the same site, the second sample is also AA. And you can summarize, actually, there'll be like 10 by 10 matrix. And we first generate this kind of matrix, and we treat this matrix as a pseudo observation. I want to emphasize here is actually this part can be pre-calculated. So that means this part has nothing to do with the evolutionary model. So you can first generate this 10 by 10 matrix. And once you want to use different kind of evolutionary model, you just from here to infer something here. So once you get this 10 by 10 matrix per pairs of samples, you can use like either GC model or GTR model to infer the pairwise genetic distance between each samples. And once you get this pairwise genetic distance between pairs of samples, you can construct neighbor join tree. This is just the first step of our method, and our future plan is to use this kind of method to infer a maximum likelihood tree. But this is the first step to generate this kind of pairwise distance tree. And the second method is basically the same thing, but I just changed this part. Previously, we assume that we know the organism we try to infer is a diploid, but suppose we don't know the ploidy level, how should we infer the genetic distance? Then actually we are not in this method, we may not rely on the genotype likelihood, because genotype likelihood, you should have a prior knowledge, whether it is diploid or triploid, this kind of thing. So we purely rely on the rate and base calling error. And if you like, you can also incorporate some kind of mapping quality score. And once you do that, in this place, based on the observation, you try to infer the joint nucleotide distribution. So let me make it clear. So suppose you got a site in one sample, it is an AT, in another sample, it is also an AT. Then actually you will say there'll be a quarter of chance to be AA, a quarter of chance you have rate AT, and a quarter of chance you have a half of chance to be AT, and a quarter of chance to be TT. So that will be taken into account in this 4x4 matrix. By doing this, actually, we don't care much about the ploidy. And once we get this, we still use the same kind of technology like the molecular evolutionary model to infer the pairwise distance, and use this one to reconstruct the neighbor joint tree. So this is basically our method. And to test our method, we try to use the previous like 5 previous methods, and also our 2, one called descends the genome, that means we know the genotype. Another one is called descends the nuke, that means we don't know the diploid or triploid, the ploidy level. And the simulation model we use to test all these 7 methods is based on this kind of thing. So we assume that this is a diploid sample, which has 2 sequences. And once you add this base calling error, and you also breaks the genotype into short reads, you will have a lot of sample here, a lot of reads here. And this is another like sample, and it is a diploid sample, and you have 2 long sequence, and you break it, adding the base calling error. And then based on this kind of thing, we realign it to the reference genome and get the genotype likelihood. Or we can purely use the base calling error and also the map quality score to reconstruct the descends the nuke method. So the standard divergence time, the standard divergence time from this tape to this tape is 0.01. And the divergence between the chromosomes in one sample is 0.004, in another sample is 0.0025. And we use this model to test our result. So first of all, I will come to the result, the method actually now people are commonly used, which is this ambiguity GT and no ambiguity GT. I didn't mention the no ambiguity GT because no ambiguity GT is you infer, suppose for this side, you infer it is a most probable genotype is a heterozygote site, and you just discard this site, and you purely use a homozygote site to construct the genotype, construct the phylogeny. And the ambiguity GT here basically means once you have the heterozygote site, you just regard it as a UPAC codes, and which will be put into the likelihood function to infer the distance. And we find actually very interesting when the rate depth is very large. This value, the 1 is always the true value, because I always, once you infer this distance, I just use the distance to divide by the true distance. So actually, if the value is close to 1, that means this method are very good. And if the value is very divergent from 1, actually, that means they are not that good. So we find it is very interesting when rate depth is large. Actually, both of these methods will underestimate the result. That is because actually they just try to infer the internal node distance from here to here. So if you really want to you calculate this kind of thing, there'll be like nearly exactly it's the same like from here to here. And when the rate depth is small, it will tend to overestimate the distance. This is because actually they cannot truly distinguish the base calling error and the evolutionary divergence. So our conclusion for ambiguity GT and no ambiguity GT is the bias are in both ways, and they can, if the divergence of the chromosome is relatively large, we cannot use that one. We cannot trust that one to infer a reliable divergence time. We now show you the result of our method and also the method of the random seq, which means per site, you just randomly choose a rate, and the consensus seq, which means per site, you just choose the consensus rate. And this is for average rate depth 0.5. And you can say actually the random seq and consensus seq will have huge bias. This is because they cannot distinguish the base calling error. And our method always stay nearly 1. And when rate depths become 5, which is the threshold, actually, I guess maybe most people use these days, like they believe this one, in this case, they can use the consensus seq method to infer the divergence time. But based on our simulation, we still say positive bias for the consensus seq. And the random seq almost stay the same because they just randomly choose one. So rate depth 5 and rate depth 1 are mathematically equivalent with each other because they just treat it as a rate depth 1. And so previous slides is for GC model. And for the GTR model, GC model is more symmetric evolutionary model, and GTR model is a more asymmetric model. And for GTR model, actually, we observe a larger bias compared with GC model. But the conclusion is nearly the same, like our method almost stay at 1, and their method have a positive bias. And we goes to the NGS model. So for the NGS model, I compare with the 2 different things. One is actually we change the distance from here to here from the range 0.01, I guess, to 1. And we do the simulation for each of the simulation result, we do 2 inference. One is for use our base dense genome model, and the other one is use the NGS-based model. And the yellow line here is the NGS-based inference result, and which the stuff actually located around the diagonal line here is our result. So our result always gave a quite accurate estimation for the divergence time. But as you can see for the NGS-based model, when the divergence time is small, you will overestimate the divergence time. And when the divergence time is large, you will underestimate it. This is because when the divergence time is large, we choose the evolutionary model to be either GC model or GTR model. The GC model and GTR model all allow this kind of recurrent mutation. But the NGS-based model, they actually assume an infinite size model. So actually, they don't allow any recurrent mutation. So when the divergence time is very large, they will tend to underestimate the divergence time. But when the divergence time is smaller, why they overestimate it? Because they do a slightly different things as what we did. What we call here is a kind of like calculate global joint distribution for the joint genotypes. But what they do here is they try to per site estimate distribution for the genotype. Then they just average them. So they average the posterior distribution for the genotypes. And by doing this, actually they cannot handle the error estimation or error reduction very well. So that's why when the divergence time is small, that means the base calling error will actually relatively contribute more. And in that sense, they will overestimate the divergence time. And here, actually, we get another interesting result. So because both of their method and our method all assert that we make use of this kind of uncertainty measurement, genotype likelihood. So then assertion should be like, when the rate depths become smaller, you should have a estimator quite that they actually the mean of the estimator should be stay the same, but the variance should be changed. Like when the rate depth is larger, you can imagine when the rate depth is smaller, you can imagine the variance to be larger and larger, but you shouldn't expect the mean change that much. But this is what we can say for the NGS-based result. So they actually change a lot when the rate depths become extremely small. But this is our result. So our result basically say the mean stay the same, but the variance become larger and larger when rate depths become smaller. So that's why I assume that, or I assert that our result, maybe our method should be better than the NGS-based. So this is another measurement. So we compare with the mean square error for each of the, for different kind of model and for different kind of method. And when the rate depth is smaller, we can at least say that our method gave a better result because the MSE is very small. And when rate depth is larger, then the consensus sake become acceptable in some sense to be used in the inference of the phylogeny. And we also use our method to infer some of the true data. So this is our method to infer ALK tree. This is totally like 83 ALK trees in, I think in the US. And we also use the other method to infer this ALK tree. And if you just look at this, if I say that our method is better, maybe I cannot convince all of you. So actually we introduce a way to measure like the good of a tree to classify this kind of known like species knowledge. Like suppose you know that some of the node of species A and some of the node of species B and some of the node of species C. And I guess if you see this kind of tree, you will say this kind of tree should be behavior, the tree inference method should be the best because 2A are clustered together, 2B are clustered together, and C are clustered together, right? And if you see the tree 2, you will say actually it is a worst one because it cannot distinguish A and B, and also C is in the middle of A and B. And the tree 3 are actually in the middle because it can distinguish B, but it cannot distinguish A and C. So we want to introduce a measurement to tell you that the first one is the best one and the second one is the worst one. This is because actually for that ALK stuff, we really don't know how long the branch is. And the only thing we know is for each node, they maybe belong to a subspecies or species. And we just want to say the topology, in the topology level, our method can do better. Then that actually say things clear. So the measurement for the topology is like, we know that for this tree, we will have like 2 internal branch, and we can cut the tree in both, in either of the branch. If we cut the tree here, it will naturally divide the tree into like 2 clusters. If we calculate the intersection, there'll be none, or there's an empty set. And if we cut in this branch and we calculate the intersection of the 2 sides, it'll be also empty side. So that means we actually introduced 2 criteria. The divide in this part, the intersection is basically 0. And basically means in this criteria, this tree pass and this tree pass. That means totally there'll be 2 branch that pass this criteria. And in this criteria, both of them also pass this criteria. That means the measurement for both of the criteria is 2. This is our first tree. And if we try to test our second tree, you'll find actually both branch will not pass first bacteria as well as the second criteria. So that means the measurement for this tree equals 0. And for the third tree, if you cut, you will find that for this more strict criteria, the measurement shows 1, and for the second one, actually both of them pass. So basically, it shows 2. So that clearly say that this tree lies in the middle of tree 1 and the tree 2, tree 2, I guess. Yeah. So now we come to the experimental data, and we actually calculate this is the first criteria, the measurement, and the second criteria, the measurement. And this is from the full data. Then we downsample it to like the average rate depth to be 10, rate depth to be 5, rate depth to be 1, like this. And you can see actually in almost all cases, our method infer the tree that go better match with the prior knowledge of this kind of species classification. Yes, with which I want to thank both of my supervisors and also my founders and also their groups for helpful discussions. And spasiba. And I would just want to like maybe take your questions. Thank you.
Thank you very much, Lei. So any questions?
Yes. Hello, Lei.
Hi.
I would like to ask one question.
Yes.
Well, it's quite simple regarding your presentation. So I would like to get a more clear picture of how exactly you incorporate genotype likelihoods into your model, and how exactly you deal with base calling errors. Maybe I just
yes
didn't get it quite right.
So actually for the genotype likelihood, it is easy because it is nearly the same way as we did with other methods. I will show you. So for the genotype likelihood, basically you assume you have a genotype likelihood, then you have an observation. You just calculate like the posterior distribution of the genotype likelihood. Of the genotype, of the true genotype. And then this actually 10 by 10 matrix is that posterior distribution. And for the other one, the nucleotide one, actually here, if you remember my teaching material the other day, actually here because I have this base calling error stuff, and what I calculate here is I always assume a GATK. That means that symmetric like genotype, how to say, the haploid genotype, actually genotype likelihood are always true. So this is a slightly different because for the first case, this genotype likelihood stuff, it can be either calculated from the GATK method or from the this Henle's method, because they are different. And also they can calculate from other maybe advanced method. But for the nucleotide model here, I only assume the A, if the rate is A and reference is C, and this chance will be a third of epsilon. And if the rate is C and the reference is also C, the probability is 1 minus epsilon, like this. I hope, do I answer your question, or do you still confused?
Yeah, I think I got it. Let me just ask another one. So did I get it correct that your baseline model is GATK?
Actually, one way you use to put this into the, like for the simulation, the model is the JATK model. And the one actually used it in practice in this 83 org tree inference. We get this VCFL, and the genotype likelihood is generated from the Henle's model. So basically, it is not JATK.
Okay, thank you.
Yeah.
Thank you once again, Lei. Unfortunately, we are out of schedule, so we do not have time for more questions. So yeah, thanks again. And now we are going to our next speaker, Sergei Abramov from Moscow Institute of Physics and Technology and Vavilov Institute of General Genetics. And Sergei will tell us about how to identify regulatory single nucleotide variants through Bayesian estimation of background allelic dosages.
Yeah.
So please.
Can you see my screen? Okay.
Yes.
Okay. All right. So as Vladimir said, I'm going to tell a bit about allele-specific transcription factor binding and how it helps to reveal some certain regulatory single nucleotide variants. If we open one of the largest databases, genetic databases, which is
Sergei, I'm afraid we cannot see some parts of your screen on the right. So there is like some black box. Yeah.
And what about now?
In this region.
What about
yes,
now?
Now it's better. Yeah.
Okay. So it's a Zoom, actually, chat box or something like this. Okay, I close it. Yeah, so if you open one of the largest genetic databases, which is Geo database, and type in human transcription factor as a search query, you'll find almost 100,000 experiments related to these terms. Well, 100,000 experiments, well, that's quite a number. But what are transcription factors in the first place? So transcription factor is a protein that can connect to the DNA, shown here, and therefore affect transcription. For example, here it facilitates the binding of RNA polymerase to the DNA, and it starts to therefore increase transcription levels. So single nucleotide, even a single nucleotide variant, can disrupt binding of transcription factor. Therefore, it will affect transcription. So here it just decreases the activity of RNA polymerase. To study transcription factors and the things, the fragments, the regions of DNA they are connected to, these regions, by the way, called transcription factor binding sites. ChIP-seq experiments are used. So in these experiments, transcription factor and DNA attached to it are immunoprecipitated using specific antibody. These DNA fragments then sequenced and mapped to the reference genome. So mapped to the reference genome, and we end up with a so-called ChIP-seq peak with transcription factor binding site somewhere inside of it. Well, surprisingly, we can study the effect of the variants in regulatory regions directly from these ChIP-seq experiments. It is known that diploid organisms like humans are diploid. So, diploid organisms like humans have two alleles, one from each parent. These alleles are pretty much the same. However, they can differ in a single letter. For example, these substitutions called single nucleotide variants. So if the variant is located, it does not affect binding of transcription factor, we would expect there will be an equal amount of reads coming from each of the alleles in ChIP-seq experiment. By contrast, if substitution affects binding of transcription factor, we expect there will be imbalance in allelic read counts coming from each of the alleles, with more reads coming from the allele with a higher affinity to the transcription factor. So if this imbalance is significant, we call such event an allele-specific binding event, or ASB. Allele-specific events are all well and good, but the question arises, why should we study them? So according to the study done in the field, around 90% of causal variants are non-coding, with around 60% mapping to immune cell enhancers, which are specific types of transcription factor binding sites. So now we have the data from the GEO database. We understand what we are trying to find, and well, it seems like all we need is computational power and like some time. However, life is never easy. So there are several complications several factors that complicate straightforward analysis of such data. So among them, the most important of them are copy number variation, reference bias, and overall heterogeneity and noisiness of the data. So I will cover all three of these topics in the next few minutes. So I start with the first one, which is copy number variation. So copy number variation is a phenomenon in which parts of the genome are duplicated or lost. So it is very common for immortalized cell lines and even for some cells of the living organism, like fibroblasts and certain neurons. So here you can see a karyotype of K562 cells. These cells are mostly triploid cells. And in this case, even if the substitution does not affect binding or transcription factor, we would expect there will still be imbalance, because like there will be 2 times more reads on average with the G letter. So in our study, we want to somehow distinguish these false positive allele-specific binding events from the copy number variation. From the genome one, I mean, yeah. So the ones that are false positives generated by copy number variation from the real allele-specific binding. So exactly for this purpose, we designed Babachi, background allelic dosage Bayesian checkpoint identification algorithm. Babachi, in a nutshell, Babachi segments the genome into the regions of constant allelic copy numbers ratio. So allelic copy numbers ratio. So let's look at the figures. Each dot here represents a variant. On the x-axis is genomic position. On the y-axis, ratio of allelic read counts is shown. So the blue stripes represent Babachi estimates. So according to these estimates, if the imbalance of a variant, for example, in this short region, should be tested against a 1/1 ratio. Sorry, meaning that we expect equal number of allelic read counts coming from the reference and from the alternative alleles. And the variant in this long region should be tested against 1/2 ratio, meaning that on average, there is 2 times more reads on one of the alleles. So in other words, Babachi helps us to find copy numbers ratios, and these ratios can be used as expected by chance proportions of read counts when performing statistical evaluation of ASBs. So that's all about SynVis, and let's talk about the next thing is reference bias. So the reference bias is a fact that occurs when we align reads to the reference genome. So if we have a variant in the genome, we have 2 types of reads. So one with the reference coming from reference allele, and one's coming from the alternative allele. The reads coming from the alternative allele have at least one mismatch. Therefore, they have a higher probability to be mapped to another genomic region. For example, bow-tie tool aligner aligns such ambiguous reads to the first entry. In other words, we expect more reads to be mapped on the reference allele than on the alternative one. This effect is called reference bias. So we decided to assess allele-specific binding significance separately for each allele, for reference and alternative alleles. To do so, we fitted two types of distributions with fixed number of read counts mapping to the reference allele and to the alternative allele. If we look at the first figure, the bar plot here shows shows the variance with exactly 10 reads mapped to the reference allele. The purple line is the fitted distribution, and we then used this distribution to assess the significance of such ASBs. So the number 10 is not important here, as we did the same for all other possible numbers. And also we did the same for the alternative allele. So in the end, every variant had two significance estimates. So one corresponding to the preference of transcription factor to the reference allele, and another to the alternative allele. So this procedure helps to overcome the reference bias because two separate distributions take into account differences in reference and alternative read mapping. Okay, so that's bring me to the last problem I want to talk about, which is like noisy data. It is known that biological data can be very noisy. So there are various sorts of biases there. For example, mistakes during sample preparation, sequencing errors, mapping errors, and etc. So many of them. However, it is a low chance that such factorations will happen simultaneously in all the experiments, at once. So to increase sensitivity of the method, we can aggregate the data. So for example, if we want to know some transcription factor specific ASBs, we can use all the experiments with this transcription factor from different cell types. Therefore, the resulting ASBs will be influenced by the properties of transcription factor rather than individual features of specific features of each cell type. And we can do the same for a cell type. So this will give us some cell type specific ASBs, which can be explained, for example, with differentially open chromatin windows. So in our study, we had more than 1,000 transcription factors and more than 600 cell types. So it means that this matrix is like more than 1,000 transcription factors and more than 600 cell types. But the matrix is pretty sparse, meaning, and we had like 10, we have median of 10 transcription factors for per one cell type. So like 10 columns per one row on average, or on median, I mean. Yeah, but nevertheless, it allowed us to increase sensitivity dramatically. So yeah, so that's all for the problems. And now I want to speak about the final outcome, the result. So the final result is Adastra database. So the final data is available online in allelic dosage corrected allele-specific human transcription factor binding sites, or just Adastra. It contains information on almost 900,000 allele-specific binding events in various transcription factors and cell types. This allele-specific, these ASBs are enriched with phenotype associations and EQTLs. Also, they are concordant with motif predictions. So the corresponding article has been already published in May this year. Here it is. And yeah, so now I want to talk a bit about how we can use this like database, this data in other studies. So for example, first example is this variant located in SMAT3 intron. So this variant is known to be, well, according to the studies, it is a candidate causal variant for coronary artery disease and Crohn's disease. The mechanism of action, the suggested mechanism of action is that this variant disrupts binding of AP1 transcription factor. So if you look at the same variant in Adastra, so this variant report page from Adastra database, we can see that Jun human is among the transcription factors, binding of which is affected by this variant. So, and Jun human is known to be a subunit of AP1 transcription factor. And if you look at the comparison of motif predictions shown on the top and Adastra data bottom, well, we can see basically that motif predictions are the same as the authors. So the transcription factor prefers to bind to the reference allele because motif p-value is lower. And the same stands for Adastra data. So as you can see, the yellow hue is a little bit is bigger than the purple one, meaning that the transcription factor prefers to bind to the reference allele. And we expect there will be 3 times more reads on average on reference allele, according to this effect size. Okay, so Adastra data, therefore, can be used to understand the mechanisms underlying some potentially causal variants. And even if this motif data is not available, the Adastra data is available for various transcription factors, and only for like 1/3 of them there were motifs available in Hokamoku database. Yeah, so example and final I want to say is speak about is Ananastra. Ananastra is another tool. It's annotation enrichment analysis of SNP sets with allele-specific transcription factor binding. So it's basically based on Adastra. And if you don't want, if you have like a list of potentially causal variants and you don't want to like search each of them in Adastra database, you can just provide them here. And well, basically tools just annotates the given list of polymorphisms with allele-specific binding events from Adastra. Well, therefore, it can help you to understand the mechanisms underlying some of the variants you provided, the polymorphisms you provided, and well, it can help to narrow your search if you search for causal variant, for example. And additionally, this tool performs enrichment analysis of provided variants with allele-specific binding events. So I encourage everyone to use Adastra and Ananastra. If you happen to try them, we will be happy to receive like any feedback. Yeah, that's all I want to say. So if you have any questions, I will be happy to answer them.
Thank you very much, Sergey. Yeah, sounds very nice. So any questions? Well, maybe you need to digest it a little bit more, but yeah,
I can like speak a little. Really I can speak about Ananastra if you if nobody wants to ask.
Just wanted to note that the names of all the instruments sound incredibly funny.
Yeah, so
I think you had a blast while creating the names.
Yeah, something like that. We were like laughing out loud with all our lab like to create these names. Yeah. Okay, so do you have time to like speak about Anastra usage, or just we are short on time and we should continue?
Well, I think you have another 7 minutes.
Okay, so but it doesn't matter actually. Okay, so actually about Anastra, when you're trying to calculate an enrichment, perform the enrichment analysis, you want to perform it against like proper background. So there are several options predefined already in Anastra. So you can use local, means that you take all the variants in one megabase around like each of provided. Whole genome means that you bring all the variants available in database and you compare with them. And OD islands, so the variants that are located in linkage disequilibrium islands for like free populations. Then there are 2 examples provided to get familiar with Anastra. So the first one is like top 500 significant variants found in COVID-19 meta-analysis. And well, there's an Anastra report page. Basically, it says about how much variance they've been found and like observed fraction and expected fraction of ASBs in the data. So there is also some odds ratio of enrichment odds ratios and p-value. So the same is done for the chromosome-wise, and also, it's not opened, but enrichment is calculated for each transcription factor and for each cell type. So you can download this like tables in download section to work with the data offline. So it can be really useful. Yeah, something like this. So thank you very much, and yeah, this brings me to the to the end of my talk. Thank you so much. So
thank you very much, Sergey.
Yeah, I think nobody have questions. So am I right? Yeah, it seems so.
So any questions? Still no questions.
Okay, so thank you.
Okay, yeah, thanks once again. It was really interesting, and yeah, the names are really cool, and it seems to be really user-friendly. It looks very user-friendly, all the interface, which is also nice. Yes, and now we are moving to the next talk by Mikhail Shishkin, the intern in the International Laboratory of Statistical and Computational Genomics in the HSD University. And Mikhail will tell us about how to infer the timing of multiple admixture events from 3 locus ancestral linkage disequilibrium. So Mikhail, you're welcome.
Yeah, hello everyone. I hope you can see my screen. Okay. So yeah, I want to talk about the method that we developed in our laboratory, the method for estimating the timing of multiple admixture events that based on 3 locus linkage disequilibrium. And estimating admixture histories is crucial for understanding the genetic diversity we see in present-day populations. And there are many methods for inferring the presence of admixture and admixture proportions. But there has been less research on estimating admixture times. And some existing approaches are based on inferring admixture tract length distribution. And over time, recombinations is expected to decrease the average length of admixture tracts. And that's why the length distribution can be informative about the time since admixture. So for example, here we have some admixture from archaic population, and red color indicates indicates loci that were inherited from archaic population. And with the next generation, because of recombinations, these admixed tracts become shorter, and we can, because of these recombinations and observing this, observing this admixture tracks length, we can tell something about time of this admixture event. And another approach that we follow in our work based on decay of linkage disequilibrium, of admixture linkage disequilibrium. And linkage disequilibrium is a non-random association between loci that exists in natural populations, but in well-mixed and genetically isolated populations with recombination, it usually decays quite rapidly at the genomic scale, but admixture tracts introduced into a population in admixture event generates admixture LD that generates admixture LD that generates much longer distances than background linkage disequilibrium. And even if the amount of LD in source populations is low. And then linkage disequilibrium in a mixed population will gradually decrease in the next generation because of recombinations. And that's why it's possible to make inference about time of admixture from the patterns of linkage disequilibrium. And for example, here we have some plots from article about method that implemented in elder software. And here we have some estimated curves of weighted linkage disequilibrium for different genetic distances. And just by fitting these curves to theoretical equations, we can estimate admixture times. And the method that implemented in elder can estimate time only to the most recent admixture event. And it uses only 2 locus LD. Okay, so we want to investigate the admixture of populations, and usually we have in this researches we have admixed population and some source populations and some number of admixture events. And in our model we have 2 source populations and admixed population. And the first admixture event when admixed population was founded by 2 source populations, and the second admixture event when the second source population contribute migrants to that mixed population. And the approach that we follow, and our approach is based on linkage disequilibrium, and LD is, like I already said, is a degree of non-random association between loci. And when a mixed population is founded, it starts with a large amount of admixture linkage disequilibrium, and then it fluctuates and decreases as a result of recombinations and genetic drift, and increases because of additional waves of migrations. In our work, we use 3 locus linkage disequilibrium, and we use Slatkin's definition, where LD of 3 loci with genetic distance d and d prime between them is covariance of this loci. And H here is represents a haplotype at locus X, Y, or Z, and it is 1 if this allele if sample carries a derived allele, and otherwise is 0. And then we can compute this covariance of haplotypes in terms of local ancestry. So AX represents a local ancestry at locus X, and it is 1 if this allele is inherited from the first source population, and 0 if it is inherited from the second source population. And consider conditional expectation, where we have Jx is allele frequency in the second source population, and delta X is differences in allele frequency in 2 source population. And if we assume that the frequencies are known and fixed, we can get next equation where we have that covariance of these haplotypes is the covariance of local ancestry multiplied by these deltas. And then we can expand this covariance of local ancestry in terms of expectation and handle it like a product of vectors. And this vector V3 consists of probabilities And the first probability is that we have one chromosome with 3 loci that were inherited from the first source population. And these 3 probabilities are that we have 2 different chromosomes, and one chromosome contains 2 loci from the first source population, and another chromosome contains another locus that inherited from the from the first source population. And the last probability is that we have 3 different chromosomes with 3 different loci that were inherited from the first source population. And these probabilities depends on generation, because every generation is formed by recombinations of randomly selected chromosomes from previous generation. And these probabilities change because of influence of additional waves of migration, genetic drift, and recombination. And we formalize this influence with linear operators, D, L, and U. And we can compute the vector for the next generation, like product of these linear operators and vector from previous generation. And this vector for generation when the mixed population was founded consists of probabilities that 1, 2, or 3 chromosome was not from the second source population. And MT here is just admixture proportion of the second source population in generation T when admixed population was founded. And now let's look at these linear operators and try to understand how can we get them. So here you can see recombination operator, and let's look at the first 2 rows. So how can we get vector for the next generation? We just can multiply, we just have a product of this matrix and vector for previous generation. So we multiply that column by first probability, plus the second column from multiplied by second probability. And let's look at these probabilities. So here we have probability for generation T. And here we have probabilities for generation T plus 1. And this probability that we have for event when we have only one chromosome with 3 loci that inherited from the first source population. So red color indicates X, green is Y, and blue is Z. And how can we get from one chromosome, only one chromosome where we have loci of the same inheritance? So we just don't want any recombination between X and Y, and we don't want any recombination between Y and Z. And probability for this event is e power minus d prime, because we model recombination as exponential distribution. And when we have 2 different chromosomes and we want to get one chromosome, we just want one recombination event here, and we don't want any recombination between Y and Z. And probability for this event is Y minus e power minus d multiplied by e power minus d prime. And the same idea for these cases. So we just don't want any recombination here and one recombination here. And when we have 3 different chromosomes, we can't get one chromosome from 3 with recombination, because recombination can occur only between 2 chromosomes. So that's why there is no any value. And when we have 2 chromosomes and we want to get 2 chromosomes, we just don't want, in this case, we don't want any recombination between Y and Z. And the probability for this event e power minus d prime. And the same here, when we have 3 chromosomes, we just want one recombination here. Okay. And now let's look at drift operator. So we just, when we just randomly select individuals from previous generation, like parents for individuals in the next generation. Let's look at these last 2 rows. And when we have only one chromosome and we want to get 2 chromosomes from one, we just should choose the same parent from previous generation. And if we have effective population size that is 2n, the probability for this event is 1 divided by 2n. And when we want to choose, to choose 2 different chromosomes as parents for 2 different chromosomes in generation T plus 1, we just should choose 2 different chromosomes from generation T. And probability for this event is 2n minus 1 divided by 2n. And we can't, and we don't have any value here because every chromosome in the next generation should have only 1 parent from previous generation. But we can choose the same parent for 3 chromosomes, and this probability is 1. This probability that you can see here, and also we can choose for 2 chromosomes the same parent, and for another chromosome just different parent from the previous generation. And also we can choose 2 different parents for 3 different parents for 3 different chromosomes. And the last operator is migration operator. It's pretty simple because it consists only of probabilities that that 1, 2, or 3 chromosomes were not replaced by chromosomes from the second source population at generation I. So here MI is just admixture proportion of the second source population at generation I. And now we can compute this covariance of local ancestry like product of these linear operators and vectors. And then if we can estimate linkage disequilibrium from real data, we can estimate admixture times just by fitting this equation. And there are some details about estimating linkage disequilibrium. So we can't directly compute LD from real data because these deltas in front of covariance of local ancestry can be either sign. And to solve this issue, we estimate weighted linkage disequilibrium coefficient which is just LD multiplied by these deltas. And the result we have these delta squares in front of covariance, and this helps us to solve sign issue. And another problem is that naive algorithm for estimating LD is very slow, and to solve this, we use fast Fourier transform to speed up our calculations. And complexity of algorithm that based on fast Fourier transform is linear in the number of segregating sites instead of cubic. And also there can be some problem when we have only 1 source population in our reference panel. So we have to estimate estimate allele frequencies for each population. But when we have only 1 source population, we miss 1 frequency for this population. So for example, we can miss allele frequencies for the second source population or for the first source population. But we still can estimate these frequencies from this linear equation, where we have that allele frequency in the admixed population equal to allele frequency from the first source population multiplied by total admixture proportion of the first source population plus allele frequency in the second source population that multiplied by total admixture proportion of the second source population. And just solving this linear equation, we can estimate missing frequency. And now let me remind you about model that we're investigating. So we have 2 admixture events. The first admixture event when admixed population was founded by 2 source population, and this event occurred T1 plus T2 generations ago, and admixture proportions for this admixture event are M1 and 1 minus M1. And also we have 1 more admixture event when the second source population contribute migrants to admixed population, And the admixture proportion for this event is M2 for the second source population. And T2 is time to the most recent admixture event, and T1 is time between two admixture events. Okay, so we did some simulations to validate our method. And here you can see simulations where we have 50 deployed individuals in each population. And each individual carries 20 chromosomes, and each chromosome is 1 Morgan length. And for these simulations, we varied admixture times and fixed admixture proportions. And you can see that when time T1, time between two admixture events, is smaller than time to the most recent admixture event, than time T2, we can see that these contours from here they predicted from theory, they are straight. And it can be hard to distinguish these cases with two admixture events from cases where we have only one admixture event. And that's why in these cases, estimation of T1 can be not so accurate. But when we have that T1 is bigger than T2, so time between two admixture events is bigger than time to the most recent admixture event, we can observe that influence of the most recent admixture event is more significant. And we can observe it by the shape of these contours, because of the shape of these contours. Okay, and also we made some simulations where we fixed admixture times and varied admixture proportions. And here you can see that when total admixture proportion increases above 0.5, these contours from theory flip from being concave down to concave up, and this satisfies our surfaces in most cases. But sometimes when a total admixture proportion is close to 0.5, we have some mismatches with theory, and we have them because in these cases variance is higher for these estimations. And also we did some simulations where we have constant admixture scenario. So constant admixture stopped 5 generations before present, and we varied starting generation and total proportion. And you can see that when we have short constant admixture, that these cases, also can be hard to distinguish them from one pulse scenario. And let's look at precision of admixture timing estimations. So here we can see estimations for T1, time between two admixture event, and T2, time to the most recent admixture event. And like I already said, when time T1 is smaller than T2, so for example, this red plot where T1 is 5 and T2 is 10, it can be, because it's hard to distinguish this case from one pulse scenario, the precision of this estimation for T1 in this case is not so accurate. But time T2, estimations for T2 are accurate for any cases. And also we applied our method to real data, and here you can see surface for weighted linkage disequilibrium coefficient for Mexican samples from 1,000 genomes project. And as reference, we have Yoruba population as source population. And we estimated for two pulse scenario, we estimated admixture times that are 13 and 8. And also we have one more example of application of our method. So here we have Colombian samples from 1,000 genomes project with Yoruba as reference source population. And here we have estimated admixture times like 4 and 14. 4 and 14. Mm-hmm. And also you can see that the difference between these cases. So here we have straight contours, and we can't be sure that there were 2 admixture events instead of 1. But here we can see that these contours are not so straight, and we can suppose that there were 2 admixture events in this admixture. Okay, so this is the end of my talk, and thank you for your attention.
Thank you very much, Mikhail. So, any questions?
Thank you for the presentation, Mikhail. Can you tell us, please, what particularly was the bottleneck, so the slow place in the naive algorithm which you boosted, like from the 3rd degree to linear in complexity?
Yeah, well, because we have to, when we have to, because we have to estimate the 3rd moment of data, if we do it naively, we should walk through chromosome 3 times, and this 3 times for every loci, and its complexity is cubic. But we can don't do that just by binning the data and computing it with fast Fourier transform as some like some oh, I forget some word. Свертка. Кто-то может мне подсказать это слово?
Convolution.
Convolution, yeah. Okay, thank you.
Thank you for your answer.
Okay, thank you very much, Mikhail.
Sorry, I also have a question. Can you hear me?
Yeah.
Okay, so my question actually is why you put the migration, actually the matrix in the front. So if you change the order of migration and drift, what happened?
If we change the order of operators in product of
yeah, because in the standard Fisher model, the migration should be happen before the drift. So that's my logic here. And the second question is, you use like this kind of like how you call it, the 3 linkage disequilibriums, or the 3rd linkage disequilibrium. And I wonder whether if you go higher order, can you get maybe more accurate result, or do you have any idea about that?
Yeah, I can ask you on the last question. So if we go higher order, we have higher variance for estimation of these covariance. But yeah, it can be with estimating higher moments of data, we can estimate much more parameters. And this now work that we do right now in our next project. But for the first question, I can't answer really for your first question, so I'm sorry.
I also want to mention, actually, for you did the inference actually to use the 3rd LD, and my previous work was actually use the 2 LD, so that's why I come to the second question.
Okay, thank you.
As far as I remember, so the operators here should be commutative, because we applied to an eigenvector.
Yeah, my previous method is also use eigenvector and eigenvalue. So I guess maybe you use the same kind of thing. Yeah.
Yeah. Yeah, so Mikhail also shows that this covariance is just that vector is an eigenvector of all the operators. So I think it shouldn't be a problem here. But thanks, we will give another look into the order of operators, at least to be sure that everything is fine. Thank you for this insight. Okay, thank you very much, Mikhail. And we are going to the last talk of our session. Our next speaker is Olesya Kondratyeva from the Institute for System Programming of the Russian Academy of Science in Moscow. And Olesya will tell us about the use of polygenic risk score for the analysis of height and body mass index in the population of Central Russia.
Hello, do you hear me?
Yes, we hear you and we see your slides.
Thank you. So, hello, my name is Olesya. I'm glad to welcome everyone who is interested in the topic of population genetics. So today I would like to present our results of polygenic risk score analysis on data of Central Russia. So when it comes to human genetics, ethnicity plays an important role because each ethnic group based on genome may have different predisposition for certain traits. There are many large-scale studies of human diseases, but for most of them, around 86% participants originated from just 4 countries. It's United Kingdom, United States, Japan, and Iceland. So when it comes to Russia, the most representative study of Russian population genetics by whole genome sequencing, to our knowledge, included just 264 individuals. That is why we wanted to find out whether it's possible to use data from other populations, and if so, how well it represents correlation between traits and genetics. So to solve this problem, we used the genome-wide association study, or GWAS. So what is that? Basically, what you do for GWAS is you take a lot of people who have certain traits and a lot who don't and who are otherwise well matched. And then you try to find place in DNA sequence where there is a consistent difference between those 2 groups. In the result, it is possible to get a score associated with every position in sequence. There are many genome-wide association studies published in open web catalog, and basically anyone can access it. In this study, we tried to estimate the degree of transferability of the GWAS result conducted on European population to Central Russia population. We used 2 traits that have been previously extensively investigated for inheritance. It's height and body mass index. We used the summary statistic from the largest to our knowledge meta-analysis of height and body mass index associated variants. We took the most valuable sets of SNPs reported by the authors of that are assumed to be independent from each other. First of all, we performed principal component analysis on our data combined with several other publicly available data sets with assigned ethnic information. In the result, the majority of our collected data were close to previously published Central Russia population. Our randomly sampled data set included people from several local minorities, but for subsequent analysis, we focused on homogenic genetic background containing people from Central Russia. Because for other populations, sample size were insufficient for profound analysis. In the study, we analyzed a total of 2,157 genomes from healthy Russian citizens, of which around 100 were used additionally to evaluate whether the analysis using European ethnic groups applies to people of Central Russian ancestry. We checked if collected genomic data has good quality metrics. Coverage of bases shown on picture A and B. On slide, on average, 19% of bases were covered with more than 10 reads. CND pictures describes called SNPs, majority of which were in previously published databases. So we can use that data set for population studies. To estimate how well GWAS represents correlation between height and body mass index and genetics, we used polygenic risk score analysis. So it is an approach for predicting personal predisposition to a certain trait, calculated by summarizing an individual risk alleles normalized according to the weight of risk allele size effect. The use of polygenic risk score improved many clinical risk prediction models for such diseases as breast cancer, type 1 and 2 diabetes. It is a fast and efficient method and can be used for human sequence data. The comparison of European and Russian cohorts based on the frequencies of previously mentioned SNPs shown on slide. So correlation coefficient for both data sets were close to 1, which points to the overall genetic similarity in terms of common SNPs, despite a clear ethnical heterogeneity among Europeans. The polygenic risk score calculation were done using Flink software, where age and sex was used as covariance in the model. So correlation between traits and corresponded scores are presented in figure on slides, where score were split into decilings. The resulting coefficient for height and BMI were 0.16 and 0.03 respectively, which closely recapitulates the results from the previously mentioned meta-analysis conducted on European population. In this study, we showed that it's possible to use studies conducted on European population to estimate Central Russia population predispositions. Similar frequency of SNPs between population allows cross-application of polygenic risk score. But it is worth to notice that recapitulation of predicting results does not necessarily guarantee recapitulation of polygenic risk score distribution across the population, And caution has to be taken in the case of transferring results to other closely related populations. So that is our studies. Thank you for your attention.
Thank you very much, Alessia. Any questions? Maybe could you repeat, please, how many individuals did you have like in your final data set?
We used around 2,000.
Around 1,000. And so all of them are like full sequence, full genome sequencing?
We used whole genome sequencing.
Whole genome sequencing, I see. Mm-hmm. Okay, interesting. And did you consider to? I think there are quite a lot of like genotyped Russian genomes. So did you consider to impute them and then also run your analysis on these imputed genomes, which would really increase the number of samples?
Well, our main goal was to estimate if we can use European studies and apply them to Russian population. So we decided to use just those 2,000. We think it's enough.
Okay. Okay. Thank you. So any other questions? And thank you once again, and thanks to all the speakers of this afternoon. And thanks everyone for coming. And I hope to see all of you soon.